Okay, guys, so now we are moving upward on our level of hierarchy. So we're now no longer dealing with the subcellular constituents. Subcellular constituents are very important. I'm sorry, let me open for you though. Everybody, please, you can turn to chapter 4. You can turn to chapter 4, and we are now moving upward in our scale of hierarchy. So back in the day, we said they used to think that anatomy and physiology was only at the level of the gross anatomy, the visible, the naked eye, which you can see with your eyes. That's anatomy and physiology. What you cannot see does not exist. But actually, they learned that the actual real anatomy and physiology, the most important essential activities are not happening at the level of the organism. They're not happening at the level of the tissue. They are actually happening at the level of not even the cell, below the cell. So we studied our macromolecules. We learned that we break down these macromolecules into micromolecules. We use hydrolysis to separate them. We use endothermic reactions to break apart these bonds. So it's very important for you. It is very important for you to appreciate what is happening below the cell that makes the cell what it is. So we don't just have a home. We have to have an air conditioner. You have to have a kitchen. So what's inside that home is what makes it a home. So the cell is the basic unit of life. It's the basic unit of death. The cell is the basic unit of health. The cell is the basic unit of disease because of what is happening underneath the cell. So now they made a level below the cell, the subcellular level, where there's atoms, where there's molecules, where there's organelles, and we discussed how we break down these organelles to get energy and how we assemble them. We use hydrolysis to break, we use endothermic reactions to break the bonds, or we join them using what kind of reaction when we join two micromolecules. Yeah, we use dehydration synthesis and composition. Excellent. We join them together, or heat-wise, we do exothermic reactions. We remove heat in order to bind them together, and that is for anabolism. That's for storage. So we learned how we store the energy, the importance of storing the energy, and now we then went on to discuss the organelles. So just having atoms and micromolecules, we have to study what's happening in the cell. We studied the membrane and what's in it. It is made up of a phospholipid bilayer. It's the big chunk of butter that has hydrophilic heads, lipophobic heads, and hydrophobic core, lipophilic core, and it has two bilayer leaflets. We talked about the ion channels that open. Some of them are gated. Some of them are leak channels. They're open all the time. Others are gated, where you need like a ligand, or you need a form of voltage change or heat. So some channels open when your skin heats up, the channels would open. Sometimes there's mechanical change. So when you feel heat, when you feel touch, all of that is because the ion channels are now open. That's how the communication, the nervous communication, the input going into the integrator, generating the output. That is all due to the action potential. The action potentials, and we are inching closer and closer to that. So the basic, simple understanding we need to have for now is that these action potentials are what mediate the transmission of information, the transmission of signal. And then we talked about the cytoplasm. And we said that the cytoplasm isn't just the liquid, that's the cytosol. The cytosol is part of the cytoplasm. But there is a cytosol, there's many organelles, like the endoplasmic reticulum, the soft endoplasmic, the smooth endoplasmic reticulum, and the rough endoplasmic reticulum. We talked about all of the mitochondria is responsible for cell life by providing energy, the powerhouse of the cell, and it is also responsible for cell life The mitochondria is responsible for cell life and cell death. And when the mitochondria kills the cell, that is known as what? Apoptosis. Excellent. Very good. Apoptosis. And a nice way of saying apoptosis is programmed cell death. Excellent. Okay, now we move on to, now we're no longer dealing with what's happening inside the cell. Now we are going to deal with what happens that generates for us and makes for us a firm, functional, homeostatic cell. And this is going to be happening in the nucleus. So the nucleus is where all the information is stored. And now we're dealing with a different type of macromolecule. I mean, we dealt in the past with fats and lipids and how they are made up of glycerol backbone and how they are made up of free fatty acids. We talked about proteins connected together by amino acids, and we talked about the sugars, monosaccharides connected together to give you disaccharides, and disaccharides connected together to give you polysaccharides. But now, those three were for ATP, for nutrition. But nucleotides now are a different, unique type of macromolecules. Themselves, they are a form of energy. You don't break them down to get energy, but you use them as a form of energy storage molecule. So nucleotides, fats are there for energy. Carbohydrates are there for energy. Lipids are for energy, and proteins are there for energy. But your nucleotides are for energy and for information. So it's like when you dial a number. If I dial a number, if you look at social security numbers, they are unique to somebody. So if I want to get to a protein, remember we began this course with the most important thing in the course, and that is the central dogma of biology. And that is we go from nucleotides DNA into nucleotides mRNA into proteins, functioning into functional or into functional. And what kind of proteins do we deal with? Functional proteins and what kind of proteins? Functional proteins and structural proteins. Thank you very much. Who can give me, what are the two classical structural, proteins that we deal with? The classical structural proteins that we deal with. So for structural proteins, keratin, excellent. Where do we find keratin? We find it in the hair. We find keratin in the nails, excellent. And we have also another type of a protein whose structural protein besides keratin. Who is that? Collagen. Collagen, thank you very much, excellent. Where do we find collagen? In the skin. In the skin and the ligament of your bone tissue. Excellent. So we said that we have the central dogma of biology, the central dogma of biology, and the central dogma of biology states that we go from DNA, who is nucleic acid, into mRNA, who is a nucleic acid, into finally proteins through translation, and the proteins are two types: structural proteins and functional proteins. Structural proteins and functional proteins. Okay, we said that DNA is a nucleic acid, mRNA is a nucleic acid, and we go from DNA with a nucleic acid to mRNA with a nucleic acid through transcription. And you go from mRNA into protein through translation. Okay, you must always begin from very big and narrow down to small. We begin with planet Earth. Then we go to the continent, North America. Then we go to the state of New York. Then we go to the borough of Manhattan. Then we locate where exactly are we. So when we want to talk about DNA, we begin very big. Obviously, it's a macromolecule. So the DNA and the RNA, both of them are macromolecules, and you can never be macromolecule unless you are made up of micromolecules. Do we agree? Unless we made up from micromolecules. So we talked in the past about the micromolecules that make up the sugars. So the micromolecules that make up the sugars are monosaccharides. So two monosaccharides will give you double disaccharides, and many disaccharides will give you polysaccharides. And we said that the sugars look like that. Oxygen closed ring with multiple carbons. And the connection is between a special type of bond, and that bond is known as glycosidic bond. That's the bond that joins for us two sugars. They could be two glucoses, glucose, and could be glucose, fructose. If it's glucose and glucose, what does that give us? Maltose. Excellent. If it's glucose and galactose, what does that give us? Lactose. And if it's glucose and fructose, what does that give us? Sucrose. Excellent. So we break this bond. We go from a polysaccharide to monosaccharide. Then we talked about the fats. The fats are made up of what backbone? What is this structure? What is this structure? What is this structure? Somebody said it. Somebody already said it. I heard it. This one on this side. And all of these are carbons. And if I want to join them together, when I join them together, what happens? If I would like to join them together, I remove minus H2O. When I minus H2O, they connect like that. When I minus H2O, they connect like that. When I minus H2O, they connect like that. So now it became a triacylglycerol. That's when they are a macro, that's when it's a macromolecule. But when I break it apart, it's gonna go back to, we add water, so OH on this side, OH on this side, OH on this side, H on this side, H on this side. We separate everything, and now it's no longer triacylglycerol. What happened? What is it now? What is this one side called? That's glycerol. That's glycerol. And what's this other? Each one of them is a free fatty acid. That's a free fatty acid. Free fatty acid. Free fatty acid. But when I combine them together with glycerol, that's now a polymer. Okay, and then we talked about the structure of the proteins. And that is, we have two amino acids. Each amino acid must have an alpha carbon, and must have a hydrogen terminal. It must have N with 3 H's, and N with 3 H's, and it must have a carbonyl carbon with OH. Carbonyl carbon with OH. So if I want to add them together, if I want to join them together, what do? I remove water, through dehydration synthesis. I connect them like that. And what kind of bond is that? That is known as peptide bond. Now we talk, so now you must be able, really able to know the structure of all of our macromolecules, how we join them, how we separate them. Now it's time to talk about the nucleic acids. So when it comes to nucleic acids are macromolecules. So the polymer is nucleic acid, and the monomer is nucleotide. Please remember, this is really, really important. So, what is a nucleotide? A nucleotide is made up of three things. I'm sorry? Okay. It's recorded, you can check it online. Okay, guys, what happens when you made me forget what I wanted to do? So it's made of 3 components. It's made up of 3 components. Excellent. We have a nitrogenous base. It's a ring that has nitrogen. That's why they call it nitrogenous base. In addition, we have a sugar that is a ribose sugar. Okay? In addition, we have a pink phosphate group. That's how all nucleic acid nucleotides look like. We can have, and the nitrogenous bases are one of 4. You're either adenine, known as adenine, or nitrogenous base. It could either be thymine, known as T, and this is A. But remember, you can never be a nucleotide unless you have all 3. So nitrogenous base and a ribose sugar, in addition to a phosphate group. Again, you gotta have a phosphate group connected to a ribose sugar, connected to a nitrogenous base. Nitrogenous base. Let me put G this time. Guanine. The amino acids, you never had to memorize all of them. But the nucleic acids, the nucleotides, there's only 4. Actually, there's 5. Because one cannot be in the RNA. RNA does not have thymine. It has uracil. So, and then you have your good old cytosine. That's a nitrogenous base. And the nitrogenous base is connected to a ribose sugar. And the ribose sugar is connected to P, phosphate. So, if you're not 3 out of 3, you are not a nucleotide. You gotta be 3 out of 3. What is the problem now if I remove one of the phosphates? So now that's a nucleotide. If I remove the purple P, phosphate, what is that? That's a nucleoside. It's not a nucleotide. So now it went down a rank. It's not a general no more. It's a lieutenant. Okay, it went down a step. So you have to remember that this phosphate group is what makes the nucleotide different from a nucleoside. So if you're 2 out of 3, you are a nucleoside. If you're 3 out of 3, you're a nucleotide. So this form right here, 3, that's a nucleotide. and we use that for information, genetic information. So we use nucleotides for genetic information. But is that the only use of nucleotides that we have? Genetic information? No. How about this? If I get adenine, that's a nitrogenous base, adenine, nitrogenous base, and I add to it a ribose sugar, and I add to my ribose sugar, I add a phosphate. What does that look like? What does this look like? Adenine, ribose sugar, phosphate. That's a nucleotide. What if I add an extra phosphate now? So now I have two phosphate. What is that now? Nucleotide still, but that extra, so now is no longer for genetic information. So now we're not using it for genetic information. That's for ATP, for energy. We use also nucleotides for energy. So now this is adenine diphosphate, right? You add one more to it, that's adenine triphosphate. So ATP is a nucleoside plus how many phosphates? 3. So if I want energy, I break this bond. Endothermic reaction, positive delta H. Or I break it with hydrolysis. Okay? If I break this bond, energy is going to come out. Plus delta G, exergonic. I get energy. This endothermic, or if I use water, hydrolysis, so energy come out. I cleave the second bond, even more energy come out. So now I go from adenine triphosphate into adenine diphosphate. Then I go from adenine diphosphate into adenine monophosphate. Then break this bond. Break this bond, and I go from adenine. When I have only one phosphate, what is that? That's monophosphate. Thank you. So when I go from adenine monophosphate into what is this? Nucleoside. So when I go from adenine, so I either go from ATP to ADP to AMP. Then my box of ATP pizza has 3 slices. I take one, I get energy. I have 2 more. I take one, I have energy. I have one last one. Then I take one, I have energy. There's nothing else. So that shows you how nucleotides can be used. And by the way, we don't only have ATP. We have GMP, guanosine triphosphate. We have CMP, cytosine triphosphate, CTP. Okay? So we have different, so these nucleotides, we can use them for genetic information, and we can also use them for energy. That's very important that you know what an amino acid is, what a fatty acid is, what is a carbohydrate, and what is a nucleic acid, and why we use them. What's the difference between a nucleotide? What's the difference between a nucleotide and a nucleoside? So I'm going to try to give you now study guide exam 2 this weekend. I'm going to send it out this weekend. So make sure this weekend, chop, finish it. You can finish the whole thing in one day, but it's going to take you time to, I told you, it's not going to stick by diffusion. You're going to keep studying it, so you can do well. Okay, so now we're going into the, and by the way, this nucleus, like I told you earlier, oh, I forgot to tell you something that's really important. Now, DNA-wise, this DNA is made up of 2 strands, right? Strand standing up. The snake is 2 snakes standing up. And the 2 snakes, they're standing up, but they are bonded horizontally and standing up vertically. So this here, imagine that strand number 1. That's strand number 2. Who's holding the strand up? That means somebody's connected. Do you think the phosphate is connected to the sugar, A? Or do you think the phosphate is connected to the nitrogenous base, B? Or do you think the phosphate is connected? Oh, nothing else. Sorry. Or do you think it is the ribose sugar? Because now we're connecting vertically. Or do you think it is the ribose sugar connected to the phosphate downstairs? That's a C. Or do you think ribose sugar connected to ribose sugar? D. Or ribose sugar connected to nitrogenous base? E. Or do you think the nitrogenous base is connected to nitrogenous base? F. Or do you think nitrogenous base connected to ribose sugar? G. Or do you think the nitrogenous base connected to this phosphate? H. These are all possible vertical bonds. You can never have any vertical bonds beside that. So if top and bottom are connected, they're connected with one of those bonds. How do you think they're connected, top and bottom? Raise your hand if you think it is A. We said A is phosphate to the sugar downstairs. Raise your hand. I need everybody, please, to participate. If you think the bond is between the phosphate and this sugar, raise your hand. If you think it's A. If you think it's phosphate and that nitrogen is base, raise your hand. Okay. If you think it's ribose and this phosphate, raise your hand. If you think it's ribose and this ribose, raise your hand. If you think it's ribose and this nitrogenous base, raise your hand. If you think it is nitrogenous and phosphate, raise your hand. Nitrogenous and ribose, raise your hand. Nitrogenous and nitrogenous, raise your hand. If you think it's phosphate and phosphate, raise your hand. If you think it's phosphate and ribose, raise your hand. If you think it's phosphate and nitrogenous, raise your hand. Our winner is there. Good job. The one-man army. Very good. It is the phosphate and the phosphate. Okay, I couldn't think of one-man thing besides one-man army. Anyways, phosphate and phosphate. That's why they call it phosphodiester bond. So now we had glycosidic bond for sugars. We had glycosidic bonds for sugars. We had peptide bonds that connect for us our amino acids. And we had these covalent bonds that bring to us glycerol with the free fatty acids. And now who's connecting for us the DNA vertically? The phosphodiester bond. Phosphodiester bond. Okay, that's number 1. That's number 1.
It's for the phosphate, so I get that. But so that's snake, and then the center that brings the nucleic acid.
I'm getting to that. Yeah.
Okay,
So and now remember the 2 strands. They are double helix. So now one strand is standing up with phosphodiester. The second strand is connected also horizontally with this green dotted line. And what I tell you about a bond when the bond is dotted, a bond is not a straight line. What does that tell you? What kind of bond is that? It's a really weak bond. Hydrogen bond. Thank you. So that's the hydrogen bond. We're not done. The hydrogen bonds between A and T are 2. While the hydrogen bond between G and C, cytosine, C, between G and C are 3. Three. Oh, that one you gotta know it. I promise you. All of this. Who are the nucleotides? Who are the nitrogenous bases? A, G, C, T. The nitrogen, how they bond? Does A bond with C, or A bond with G, or A bond with T? A bonds with T. A bonds with T with how many bonds? A bonds with T with 2 bonds, and G binds with C with 3 bonds. Vertically, We are connected with a phosphodiester bond with two phosphates, and horizontally we're connected with a hydrogen bond. We're not done. These nitrogenous bases are put in two groups. If we say pure or good, pure or good. So that means if you are a purine, if you are a purine, if you are a purine, that means you're adenine, A, and guanine. Pure or good. Pure or good. You are purine. But if you are not A and G, which means you're either a T or a C, and if you are in RNA, RNA has no T. That one, you got to know that one. That RNA does not have a T. RNA has a U, uracil. And those three are not purine, is pyrimidine. Pyrimidine. So many students think that pyrimidine, because it has a long name, has the big one. No, the purine are bigger than the pyrimidine because they have two rings, two circles we're gonna see, while pyrimidine has one ring. So the one with the longer name has the most letters. The G, the C, and I'm sorry, not the G. The C, the T, and the U, cytosine, uracil, and thymine are pyrimidines, while adenine and guanine are both your purines. Very important. Which one has two rings? The big one, the purine. Pure or good, adenine and guanine. Okay, guys. Alrighty then. Okay, any questions so far? Okay. Now, another thing. Oh my God. The sugar, the red one, the red sugar. The red sugar in the DNA on its 2 position, it has a 2' position. On its 2 position, it is not an OH. The O is gone. O is erased. So it's deoxy. Devoid. It was removed. So it's deoxy. That's why they call it DNA. Deoxynucleic acid. DNA. Deoxynucleic acid. You got it? But in RNA, that O is there. So it's still, we call it ribonucleic. We don't call it oxy. If you're normal, you are ribonucleic. If the O is gone, you are deoxy. That's why we say DNA and RNA. So RNA, ribonucleic. DNA is not, the sugar is a ribose sugar. We said that one already. Red is a ribose sugar. But the ribose, does it have an oxygen? You are just a ribose. If you are a ribose with no oxygen, you are a deoxyribose. And the DNA is the deoxyribose. Okay, now let's go through the slides. Deoxyribonucleic acid DNA is long thread-like molecule with uniform diameter but varied length. There are 46 DNA molecules, chromosomes, in the nucleus of most human cells. The average human DNA molecule is about 2 inches long. The average human DNA molecule is about 2 inches long. DNA structure and function. The DNA and other nucleic acids are polymers of nucleotides. They are polymers. We said that. Each nucleotide consists of 3 components: a sugar, deoxyribose, a phosphate group, a nitrogenous base. So there's a DNA bases A, T, C, and G. Purines, you see the purines? Purines, say pure or good. Purines, adenine and guanine. They have a double ring structure. While the pyrimidines, they are cytosine, thymine, and uracil. They have a single ring structure. Oh, what does this look like? What does this look like? Red, green, and yellow. 3 out of 3. 3 out of 3.
Hold on, just oh, you said the guanine up here on the board is a triple one up there.
I'm sorry?
See, there's 3 lines on the board.
Here?
No, on the board. And green.
Yeah, between guanine and cytosine is 3 bonds. They bond like that. When they are like this, they bond with 3 bonds. But if it's A and T, they bond with 2 bonds.
But here it says
Here where? Here where? It's fine. Okay. Now, what do we have? We have 3 out of 3. What is red? Adenine. But what is it? It's a nitrogenous base. How many rings does it have? 2, because it's a pyrimidine or purine? Pure. Pure are good. The one with short letters is the one that has the big rings. 2 rings. Now, let's look at the ribose. Look at the 2' where my mouse is. You see the mouse? Now, the mouse here is supposed to be just like this one. It's supposed to have an OH, but the O is gone. So it's deoxyribose, but still a ribose, but it's deoxyribose. And that's a phosphate. Now, that's a nucleotide. If I add 2 more phosphates, this is no longer going to serve as a DNA nucleotide, but it's going to be a form of a nucleotide, not for information, but for what? Energy. The 5 nitrogenous bases found in DNA and RNA. You see the purines? Purines are adenine. It has 2 rings. You see the first ring, second ring, and the guanine has purple ring and purple ring. Now let's look at pyrimidines. Pyrimidines are your cytosine, thymine, uracil, and they have the longer name, but they actually have only one ring. Structure and function. The double helix shape of the DNA resembles a spiral staircase. So each side piece is a backbone of phosphate groups alternating with deoxyribose. So the step-like connections between the backbones are base pairs. So nitrogenous bases are united by hydrogen bonds. They are united by hydrogen bonds. And we talked about that. I'm sorry? Okay, purines pair with pyrimidines where A binds with T with 2 hydrogen bonds. That's all money. That's all business. A binds with T through 2 bonds, and G binds with C through 3 bonds. The law of complementary base pairing. The law of complementary base pairing. So there's a sequence of one strand that governs the sequence of the other. So we know that if you are A, you will definitely have to bind with the T. Or if you are A, you would bind the U if we were in RNA. So that's the double helix. Can you please examine this structure? If you look at this structure, first of all, I said we begin big, then we narrow down. That's very big. That's the DNA. Now we narrow it down into what? Into 2 strands. 1 strand, 2 strand. Then hold 1 strand. It is made up of nucleic acid. How many nucleic acids are here? Not nucleic acid. The nucleic acid is the whole polymer. But I'm talking about the monomers. We have 1 nucleotide here, A, green, the yellow on top, not the yellow on the bottom. Okay? Red, green, yellow. Red, guanine, sugar, deoxyribose, sugar, phosphate. And they are connected through this phosphate bond. So there's 2 oxygens here are connected by this, are connected through this phosphate group. The other thing you need to know is that our DNA is what charge? It has a phosphate, just like the bilayer leaflet. It has a phosphate. What charge is phosphate? What charge is phosphate? Phosphate is minus, so that means the plasma membrane is minus. Excellent. And that makes the DNA what charge? Negative, because of the phosphate. Excellent, 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 excellent. So that one is very important. And then there's the complementary base pairing. And then there's the polarity. If the P is the 5' position. So the yellow, that's the 5'. P represents the 5. And OH represents the 3. Because remember, in the sugar, you had a deoxyribose at what position I said? What position did we say in the sugar? It was that when there was no OH or OH. Where was the sugar? On what position? Look, that is carbon number 1. That is carbon number 2, 3, 4, 5 at the top. So the problem is here, number 2. But that's number 5. If you go down to the DNA structure, what do you see here? So that's carbon number 1. That's carbon number. That's the one that tells us, are we looking at DNA or RNA? How do you tell if we're looking at DNA and RNA? What's happening at carbon number 2? There will be an oxygen if we are RNA. Excellent, excellent, excellent. Then 2. Then position 3 is what tells us the polarity in the DNA. So this position, that's 3. And the phosphate is bound to carbon number 5, right? Phosphate binds here. Here, you see the yellow phosphate? The phosphate binds to which number? Does it bind to 2? Does it bind to 3? 4. It binds to 5. So that means the 5' position is right here. So that shows you this DNA, that's the 5'. Now it's going, this is 5', that's 3'. Look here, this is now the 5' on the bottom, and that's the 3' on the top. So they...
Professor, what are you counting?
Okay, please look there, look there. You see this sugar in the middle, right? This sugar, you see this sugar? This green sugar. It has how many carbons? 1, 2. The dot is a carbon. The dot represents, in the organic chemistry, that represents a carbon. So that's 1 carbon, but not this dot. That's an N. If it's an empty dot with no letter next to it, that's a carbon. But this one is, you see, this dot has a hydrogen, so that represents a hydrogen. But this intersection here, that is a carbon. But that's carbon number 1, carbon number 2. Carbon number 2 is the one that tells us, are we with oxygen? That makes you ribose. If no oxygen, that makes you deoxyribose. That's number 3. That's number 4 in the corner. That's number 5 at the top. So this 5 at the top is where we add our phosphate. So now when you look at the DNA, the same thing. Look, please look there. Look, please look there. Look at my mouse. This is the carbon number 1. That's the carbon number 2 with the OH. That's the carbon number 3. That's the carbon number 4. And that's the carbon number 5 attached to my yellow phosphate. You see, that's carbon 5 attached to the yellow phosphate. So that's the carbon 5 attached to the yellow phosphate. So that is the 5' position, because it's attached to the 5th carbon of this ribose sugar. Okay? But the bottom one, look, that's 1. Count with me. What number is that? What number is that? What number is that? Because the OH here, this is the OH. So the 5' position represents this phosphate with this 5 carbon. And the 3' positions represent the OH who's bound to this 3rd carbon. So DNA has a 5' and a 3', representing that you have a phosphate bound to the 5th carbon, so 5 up top, and here to the bottom, that is the OH bound to the 3 carbon. So now the complementarity is we are 3 going 5. On the opposite, you will see that the strands are like two snakes, but one has its head up, its tail down, and the second one has its head down, its tail up. You can tell. So you tell me. Let's begin with the T. The T is bound to which number? Is it number 3 or a number 5? That's the number 3. So it is bound. So it is not bound to number. It's bound to number 1. You see, we bind the nitrogenous base always at number 1. it is the 3 and the 5 are here. Are you bound to a phosphate, or are you bound with 3 to downstairs with the other phosphate? So you see it like right here. That's our number 1 because it binds the nitrogenous base. So you gotta respect all your carbons. The first carbon, carbon number 1, binds the nitrogenous base. The second carbon is the one that is responsible for telling us is there deoxyribose or oxyribose. The third one is so this phosphate connects 5' to 3' sugar. So let's look here now. That's nitrogenous base, so it's always connected to what number? 1. That's number 2. That's number 3. So this is our third carbon bound to the phosphate. That's number 4. That's number 5. So 5 is bound to the phosphate, but the phosphate of the second one binds to the carbon of number 3. So the phosphodiester bond is between who? The phosphodiester bond is between the 2 sugars, but between position 5 and position 3. You see, if you have 2 nucleotides like that, you have the phosphate binding the top at number 5. You see that's carbon number 5. You see my mouse? That's carbon number 5 bind to the phosphate, and then the phosphate binds the down, not at number 5 again, but at number 3. So you got to know the significance of each carbon. The first carbon binds the nitrogenous base. Second carbon tells us, are we deoxyribose or ribose? Carbon number 3 participate in the phosphodiester bond, carbon number 5. So these are the most important ones. The useless one is number 4.
Professor,
Yes.
there's no oxygen over there though, so how does that tell you that it makes it oxygen?
Because there is no oxygen there. It's deoxy. Why is it deoxy? D is devoid because there is no oxygen. But when we get to, now this is the structure of a DNA nucleotide. That figure is confusing because it doesn't say the structure of a DNA nucleotide. But if it was a structure of an RNA nucleotide, the H is going to have an O. Okay, so that's just for you to know that if you have the phosphate group at the top, you know that's the 5'. That's the 5'. But if you have the OH at the bottom, then that's the 3'. And the snakes are parallel, but one has a head up, tail down. The other one has a tail up, head down, and they are bonded. Okay. The essential function of DNA is to carry instructions, genes, for the synthesis of proteins. So the gene is a segment of the DNA coding for the synthesis of a specific protein. So humans have about 20,000 genes, only about 2% of the total DNA. The other 98% is non-coding DNA. It plays a role in the chromosome structure. Okay, so we already talked about the central dogma of biology. We said that the central dogma of biology is important because it shows us the flow of information. We go from nucleotide DNA to nucleotide mRNA into the protein. Excellent. Which is not a nucleotide. Proteins are amino acids. So the DNA is organized with proteins known as chromatin, and the chromatin are the fine filamentous DNA material complex with proteins called histones. So histones are like coils. We bind them together. We bind these coils together. And these coils are very important because if the DNA is negatively charged, that means in order to stick together, what charge they gotta be? For the histone. If the DNA already has a negative phosphate group, what will the histone have? Okay, chromatin. So the DNA is organized with proteins, and it's condensed. So the reason why it's condensed is because of these coils. So I had that for you in that illustration. I'm going to play it for you in the end. So we're going to watch it as a class now. So now no headphone issue. Now all of us, we're going to watch it like a movie. So the chromatin is condensed in these coils. These coils, they are fine filamentous DNA material complex with proteins called histones. Complexed with proteins known as histones. It occurs as 46 chromosomes in most cells, and 6 feet. They are 6 feet long, and you see this it is so condensed because it is wrapped around the histones. So the DNA double helix. The DNA double helix is you see now you see the two snakes, and now you see that they are connected by hydrogen bonds. So one is the DNA double helix, two is the DNA winds around the histones to form the core particle. And then you have chromatin folds into a thicker zigzag fiber. And then the fiber folds still further into irregular loops with increased thickening. Okay, so in dividing cells, looped chromatin forms into thick chromatids visible to light microscope. And the chromosomes at the midpoint, the metaphase, these are the chromosomes at the midpoint metaphase of the cell division. So histones, that's very important. Histones are proteins around which the DNA winds, and histones cluster in groups of 8 molecules. So DNA molecule winds around the cluster like a thread around a spool, and it creates a granule called a core particle. So chromatin consists of thousands of repeating core particles. So chromatin forms complex loops and coils, 150 times thicker and 1,000 times shorter than the naked DNA. So each chromosome is packed into its own region of the nucleus called a chromosome territory. It's permeated with channels allowing regulatory chemicals to have access to the genes. So in non-dividing cell, the chromatin is not elastic. So the structure and location changes moment to moment. So according to the genetic activity of the cell, genes get turned off and on. So when preparing to divide, the cell makes copy of all the nuclear DNA. So each chromosome then consists of 2 parallel filaments of identical DNA called sister chromatids. And the sister chromatids, in their own structure, they have that is very similar to the hemoglobin. In hemoglobin, you have the heme group, and the heme group has an iron group, and the iron group attaches to the oxygen. So here the sister chromatids in the middle are joined together with centromere. But the centromere has a protein in front of it known as kinetochore. So these protein plaques on each side of the centromere play a role in cell division. So chromosome structure at metaphase, that shows you the chromosome. So this is the centromere. You see the centromere is the middle line, and these orange pieces are your kinetochores. Now we're gonna move to ribonucleic acids. So ribonucleic acids come in many forms with diverse functions. So the general structure, they contain the sugar ribose. And the bases are adenine, uracil, guanine, cytosine. Uracil replaces thymine found in the DNA. So there are single nucleotide chains, not a double helix, except in short regions. So smaller than DNA, and they can have less than 100 or just over 10,000 bases per molecule. And it functions mainly in the cytoplasm. There are 3 RNAs that you have to know. These are the majors RNAs. They're not the only ones. I had an illustration, a demonstration that is very important on YouTube about microRNAs. That is a form of an RNA. Small non-coding RNAs, snRNA. That is not a major form of RNA, microRNA. Okay, small non-coding RNA. But the ones that you need to know are the messenger RNA, the transfer RNA, and the ribosomal RNA. Who can label those 3 for me in order of ascending order? Which one is the smallest of those 3? The transfer RNA, the ribosomal RNA, or the messenger RNA? Ribosomal. It's the tRNA. It has only 3 codons. It's so small. Ribosomal RNA and messenger RNA, who's the bigger? Nope. The messenger is about sometimes 10,000. I used to think the ribosomal, because it's part of the ribosome, is a ribozyme, but not really. It is.
Okay.
Hmm?
Sorry, so sorry. So it's transfer, ribosomal, and then messenger?
Yeah, it's labeled already.
Right.
You see. Yeah. Okay. All right. So now we're gonna talk about genes and their action. So a gene is difficult to define. So the previous classical definition of a gene is a segment of a DNA that carries the code for a particular protein. But the body has millions of proteins and only about 20,000 genes. So the current definition is a gene is an information containing segment of the DNA. So it has to be in the DNA. If you are a gene, you gotta be in the DNA. That codes for the production of molecule of RNA that most often plays a role in synthesizing one or more proteins. So the amino acid sequence of a protein is determined by the nucleotide sequence in the DNA. The amino acid sequence of a protein is determined by the nucleic acid sequence of the DNA. So the 46 human chromosomes. The genome refers to all the DNA in one 23 chromosome set. So about 3.1 billion nucleotide pairs in the human genome. So single individual variation comes from single nucleotide polymorphism. What is the major single nucleotide polymorphism you always need to know? There is one single nucleotide polymorphism, no matter what course you're gonna get into, you have to know it. It's an SNP. It's the one that leads to sickle cell anemia. That one you have to know it. The single nucleotide polymorphism is when we change from glutamic acid into valine. And then because of that, when there is a demand for oxygen, the red blood cells start to sickle. So remember, they're going to ask you, in sickle cell anemia, what amino acid were you supposed to have originally? Is glutamic acid. When a mutation happens, when a single nucleotide polymorphism happens, that glutamic acid is still there? No, it's not. Because a mutation happened, another amino acid came in its place. What is the amino acid, the second amino acid that came in the place of glutamic acid? It's valine. So that single nucleotide polymorphism that changed for us, glutamic acid into valine, led to sickle cell anemia. Our red blood cell in sickle cell anemia sickle all the time? No, only when you need oxygen. So when you stressed out, when you exercise, or when there is drop in oxygen level, they start to sickle. So when you need them the most, that's when they start sickling. Genomics is the study of the whole genome. That's very interesting because now they realize there's something known as genomic medicine. What is genomic medicine? Genomic medicine is when you study the DNA and you see that there are regions that are always mutation, like this one, like the sickle cell anemia. So they study the DNA regions that are usually undergoing mutations over and over, no matter who you are, no matter what country you're from, no matter what male or female, that region is always a lot of problems with it. So they study it a lot and do research in it and focus therapy on that region. So this is known as genomic medicine. So the application of knowledge of the genome to the prediction, diagnosis, and treatment of disease. It is relevant to many disorders. Examples are cancer, Alzheimer's disease, schizophrenia, obesity, AIDS, and tuberculosis. So the body can make millions of different proteins. The proteome. The body can make millions of different proteins. The proteome. So from just 20 amino acids and encoded by genes made of just 4 nucleotides. ATCG. The genetic code is a system that enables these 4 nucleotides to code for the amino acid sequences of all the proteins. So the base triplet is the sequence of 3 DNA nucleotides that stands for 1 amino acid. So the codons, you have 3 base sequences in the mRNA. So in the codon, you have 3 base sequences in the mRNA. There are 64 possible codons available to represent 20 amino acids. So that shows you the DNA is redundant, meaning that different combinations of amino acids can give you the same, different combinations of codons can give you the same amino acid. That means that this is redundant. So the genetic code enables these 4 nucleotides to code for the amino acid sequences of all the proteins. So the base triplet is a sequence of 3 DNA nucleotides that stands for 1 amino acid. And in the codon, you have 3 base sequence in the mRNA and 64 base codons available to represent 20 amino acids. 61 code for amino acids and 3 are stop codons. So there are so you need to know the stop codons and the start codons. The stop codons, you got to know the 3 of them. You are gone, you are away, you go away. UGA, UAG, UAA. You go away, UGA. You are away, UAA. You are gone, UAG. These are your 3 stop codons. You got to know them 100%. Because there's a question could tell you which of the following is a stop codon or which of the following is not a stop codon. Very easy. You tell someone, you go away, that's UGA. You are away, UAA. And you are gone, UGA. Those 3 are your stop codons. Renee, what are the 3 stop codons? That's not a stop codon, you go away.
Oh, stop codon?
Yeah.
Stop?
Yeah.
Oh, sorry,
Yeah. So you can, yeah, no, you can't say that. You just have to give me the first letter, but it helps you to memorize, I know. But you gotta know it. And you know, and you gotta know also your start codon. AUG. So AUG codes for methionine, and begins the amino acid sequence of the protein. That's methionine. Yes. Okay. You go away. UGA. You. Yeah, UGA.
Go away.
Yeah, I'm sorry. I'm trying to tell her how to remember it, but I'm not trying to tell what they are. You go away. No, they're random. But if the ribosome sees one of them, stop the track. It's gonna stop. It's like a stoplight. We finished. We finished the translation. Yeah, with one of the three. So UGA, you go away. UAA, you are away. And you are gone. UAG. Okay. What is the start codon? That's AUG. And you gotta remember that it is 2 in 1. Buy one, get one free. It's a start codon and it's an amino acid. That's a very high yield one because you get asked the start codon. The stop codon code for nothing, just to stop. But the start codon, it start and it will give you an amino acid. What is it? Methionine. 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 Okay, protein synthesis. So the big picture. So all the body cells, except the sex cells and some immune cells, contain identical genes. All the body cells, except the sex cells and some immune cells, contain identical genes. So different genes are activated in different cells. That was an amino acid that gets coded by a start codon. So different genes are activated in different cells. So any given cell uses 1/3 to 2/3 of its genes. The rest remain dormant and may be functional in other types of cells. So when a gene is activated, messenger RNA is made. It serves as a code for a particular protein. So going from DNA to RNA to protein. So DNA to RNA is called transcription. And that occurs in the nucleus. And the mRNA to protein, that is called translation, and that occurs in the cytoplasm. Now we're going to talk about transcription. So transcription is copying the genetic instructions from DNA to mRNA, copying the genetic instructions from DNA to mRNA. And it utilizes enzymes called the RNA polymerase, and it binds to start sequences in the DNA and it opens up the helix. So it reads the bases from one strand of DNA to build a complementary strand of mRNA. So where it finds C on the DNA, it adds G to the mRNA. Where it finds G on the DNA, it adds C to the mRNA. Where it finds T on the DNA, it adds A to the mRNA. But where it finds A on the DNA, it adds U to the mRNA. That is very, very important. So what happens in transcription is that RNA polymerase is going to copy whenever it reads on the DNA. When it reads the C, is gonna make the complementary strand, and that will be a G. If you have a G, if it sees a G, it will make a C. And if it sees a T, it will make an A. But if it sees an A, the RNA polymerase is a ribonucleic acid. So it does not deal with thymine. It's going to put uracil, a U. So we do not need, so this RNA polymerase is DNA dependent. I hate how they don't put that on the slide because it is reading off of DNA. So its name is not just RNA, it is DNA dependent RNA polymerase. Because who is the wicked one who uses RNA dependent RNA polymerase? It's an RNA dependent DNA polymerase. Okay, think of it this way. If I'm going from here, if I'm going from DNA to mRNA, I read the DNA and transcribe it into RNA. So who's doing that? It's the RNA polymerase. But who is it dependent on? It's dependent on the DNA. So that makes it the DNA dependent RNA polymerase. Now remember the HIV virus? What I tell you about the HIV virus? It reverses what? What does transcription? So it goes from RNA to DNA. So what is it? Is it an RNA polymerase? No, it's a DNA polymerase. Is the DNA polymerase? It's a DNA polymerase. And what is it dependent on? What is the reverse transcriptase dependent on? It is dependent on the RNA. So that makes it a RNA dependent DNA polymerase. It makes DNA from RNA. But if you make RNA from DNA, that makes you DNA dependent. I heard wait somewhere. Did I go too fast? Okay. Yes. Okay. Think about it this way. I gotta depend if I want to transcribe. If you want to scan something, you gotta have the actual template. So if I want to make RNA, I depend on DNA. But what am I making? I'm making RNA, so I'm an RNA polymerase. Who am I dependent on? DNA. So I'm a DNA dependent RNA polymerase. But the virus, the HIV virus, it doesn't have DNA. It does not have DNA. It only has RNA. But what it does, it actually doesn't even have RNA. It has mRNA. It has mRNA. What it does is it reverses to make DNA. So does it have a DNA polymerase or an RNA polymerase? RNA, if you make an RNA, you have an RNA polymerase. When we're transcribing mRNA, we need polymerase means what? It means it makes polymers. So RNA polymerase means it makes polymers of RNA. But if I'm going with reverse transcription, if I'm going from RNA to DNA, what polymer I'm going to use it? DNA polymerase. And who am I reading? Who am I dependent on? Am I a DNA dependent DNA polymerase or I'm a RNA? Say it, I'm a RNA what? RNA dependent DNA polymerase. I make DNA because I depend on the template of the RNA. In replication, in the cell division, when I double up, I'm not going to no RNA. I have DNA. I make another copy. I make double copy, 2x. So I have DNA. Now help me tell, what kind of polymerase, who am I dependent on? If I make DNA from DNA, what is that? What polymerase is that? That's a DNA polymerase because it makes DNA polymer. DNA polymerase. And who am I reading off of? DNA. So that is DNA dependent DNA polymerase. And that is only in replication. When we are replicating in cell division, we double up the DNA in the synthesis phase, known as the interphase. Okay? And then we prepare to go to mitosis so we can split up. Okay? Transcription. So the RNA polymerase rewinds the DNA helix behind it. It allows the gene to be transcribed by several polymerase molecules. So it's like a gang A big gang, each one has a job. So when we want to replicate, we want to transcribe, the gang comes. The first one says, I'm going to unwind this double helix, these two snakes who are connected. I'm going to unwind them. I'm going to remove this tightness. Then the second one says, thank you. I'll take it over from there now. It's a teamwork. The second one, gang work sometimes is teamwork too. It's a negative teamwork. So they open up the helix. One opens up the helix. And the second one, but this is good teamwork. They open up the helix and one start to transcribe the DNA. And the other one starts to put as we will see all of their steps. But it's a teamwork. It's a gang work. It is not just one enzyme who's doing the job. So let's see here. So the DNA polymerase rewinds the DNA helix behind it, allows the gene to be transcribed by several polymerase molecules. So obviously, translation happens at the ribosome, but it's not just one ribosome who is working. It's multiple ribosomes transcribing the same mRNA. And when we are transcribing, yes, the DNA, the RNA polymerase transcribe, but it is not the only RNA polymerase transcribe. You have multiple ones transcribing. Allows genes to be transcribed by several polymerase molecules. And there's a terminator. So the terminator stop is a stop sequence. So now the same way we had a stop codon, but that stop codon was for translating proteins. Now we're talking about transcription. So there's a stop sequence to stop at the end of the gene to tell the polymerase, stop. You don't need to transcribe nothing no more. You have enough template that can be translated. Pre-mRNA is immature. Pre-mRNA is immature RNA produced by transcription. Pre-mRNA is immature RNA that is produced by transcription. It undergoes processing. And the enzymes within the nucleus remove introns from the RNA and splice exons together. So the pre-mRNA is immature, and the RNA produced by transcription undergoes processing, and the enzymes within the nucleus remove the introns from the RNA and splice the exons together. So this mRNA that we make from transcription, this is known as the nascent mRNA transcript. What does nascent mean? What does a nascent transcript mean? Nascent mRNA transcript. It means that it needs to go more surgery. It needs to have more cutting. So I'm going to cut regions, throw them out, and regions keep them in. So the regions that I'm going to throw out are called introns. Students mix that up. They think an intron because it has an in it, an intron, and an exon because it has an X. They think that, oh, an intron is going to remain in the mRNA and the exon is going to get exited out. No. The exon is going to stay and the intron is going to be thrown out. So think of it this way. Where is transcription happening? Inside the nucleus. So when I cut things and throw them away, they stay in the nucleus. So they are introns. They're not going outside. It is the exon who's going to exit the nucleus now, and it's going to go to the ribosome for translation. So that's the exon. So the exon, it means we need you. So you can exit the nucleus and go to the side because DNA is so big, it cannot leave the nucleus. But we can make this exon, and the exon is the part of the mRNA that will go. What about the introns that we stay behind? You know, we always love recycling. We talked about that already, about how when we have superoxide, we have the enzyme superoxide dismutase that muted down into hydrogen peroxide. Then the enzyme hydrogen peroxidase comes in and converted into water and oxygen, or catalase comes in, breaks it down into something that's even more useful. So we're gonna recycle this intron. So all of these all of these not needed pieces are gonna be joined together, and they're gonna become later on mRNA. This is called alternative splicing. Alternative splicing. So all the stuff that I cut out and stays behind is not an exon. Is not good enough to be exon to leave the cell, to be transcribed, to be translated there. No, no, no. We're not going to garbage. We're going to assemble you guys together and make something out of you. That's called alternative splicing. Oh, right there it is. Alternative splicing. You guys see alternative splicing? Is the variations in the way exons are spliced allow for a variety of proteins to be produced from one gene. So one gene can code for more than one protein. Exons can be spliced together into a variety of different mRNAs. So the exons are going out. So it's no longer a nascent. So the nascent, what is the nascent mRNA transcript? Yours. Yeah. It's both. Is when you have the good and the bad together. What I need, what I don't need. But when I splice out the exon and send it out, then I don't have nascent no more. That's a mature. It's modified now. Now it's ready to be transcribed. Okay? And then the introns that stay behind, what do we do? You stay, they just pile up there? Yeah, we alternatively assemble them together and make them into useful exons. Excellent. Oh, there we go. So that's my gene. I do transcription, and that's my, you guys see the illustration that says pre-mRNA. So this pre-mRNA is going to be spliced. You see all the brown parts? We do splicing. We cut it out. We removed all the introns. Then we connected all the exons, and then we have our proteins of interest. When it comes to translation is nucleotide language converted into amino acid language. And the ribosome reads the mRNA code and forms a protein. So there are three main participants in the translation. There's the messenger RNA that carries the code from the nucleus. It needs to be read. It has protein cap. Okay, it has poly A tail and 5' G. It has a G cap. It has a G cap, guanosine cap, 5' guanosine cap, and it has poly A tail at the 3'. That's the messenger RNA. Then the transfer RNA is the tRNA. It delivers a single amino acid to the ribosome, and it contains an anticodon. Series of 3 nucleotides that are complementary to the mRNA codon. Ribosomes. Ribosomes are organelles that read the message that build a peptide chain. They contain a large subunit and a small subunit. So free in the cytosol on the rough endoplasmic reticulum and on nuclear envelope. So it's free in the cytosol on the rough endoplasmic reticulum and on the nuclear envelope. What do we say about the ribosomes that are free floating and the ribosomes that are embedded on the rough endoplasmic reticulum? Both of them make proteins. What's the difference in the proteins they make? Very good. The free floating they don't make them for the nucleus. They make them for the cell. Because what are we making? We're making proteins, and proteins, they don't only help the cell. You know those membrane channels, what are they made of? Plastic? Proteins. So we need proteins for cell membrane. We need proteins for the cytoplasm, proteins for the nucleus. So now the ribosome has 3 sites: an exit site, E site, and an A site, and the P site. So the E site is how we leave the cell. And it is made up of large subunit that is about is made up of one is a 40S and a 60S and an 80S. We will see in a little bit. So that's the amino acid accepting end. That is the anticodon. So there are three main steps in translation. The first one is known as initiation. And in initiation, the ribosome assembles with mRNA in the cytosol. Small subunits bind leader sequence of mRNA, and large subunit joins when AUG. Who's AUG? Who is AUG? Start codon. What amino acid does AUG have? Methionine. Excellent, excellent. So what happens is the ribosome assembles with the mRNA in the cytosol, and small subunit binds the leader sequence of the mRNA, and the large subunit joins. When AUG codon is reached, protein synthesis begins. tRNA brings first amino acid methionine. Okay, elongation. So in elongation, the next tRNA with amino acid arrives. It binds to the A site. The ribosome creates peptide bond between the first and the second amino acid. The ribosome moves down the mRNA by one codon. So growing peptide is now attached to tRNA in the P site, and the process continues and the peptide grows. Termination is when the ribosome reaches a stop codon. So A site binds a protein called a release factor. The ribosome disassembles and dissociates from the mRNA. So making proteins for packaging or export, we talked about that if we want to make proteins for exporting, then the proteins destined for the lysosome or secretions are modified by the endoplasmic reticulum. So the ribosome docks in. So the ribosome docks inside the endoplasmic reticulum and synthesizes protein into the endoplasmic reticulum sister. And the endoplasmic reticulum modifies and packages the protein into transport vesicles. There are multiple ribosomes in clusters called polyribosomes, and they can translate the same mRNA molecule simultaneously. So the protein cap, the ribosome subunit binds to the, so there's a protein cap and the pool of free amino acids. So the tRNAs bind an amino acid and they give us proteins. Okay. This is a polyribosome. And the DNA is a double helix. There are 7 base triplets on the template strand of the DNA, and the corresponding codons of the mRNA are transcribed from the DNA triplets. So each triplet is read, and then an anticodon is generated. And then the anticodons of the tRNA bind to the mRNA codons, and the amino acid carries by the 6 tRNA molecules, and the amino acid is linked to the peptide chain. Protein processing and secretion. So protein synthesis is not finished when the amino acid sequence, the primary structure, has been assembled. To function, proteins must fold into precise secondary and tertiary structures. So there are chaperone proteins. These chaperone proteins guide the folding of newly synthesized proteins. So these newly synthesized proteins help prevent improper association between different proteins. So some also call stress proteins or heat shock proteins, and chaperones produced in response to the heat or the stress. They help damaged proteins fold back into the correct functional groups. So number 1, the protein formed by the ribosomes on the endoplasmic reticulum. And the protein is packaged into transport vesicle when it buds from the endoplasmic reticulum. And then the transport vesicle fuse into clusters to form a new cister. And the Golgi apparatus modifies the protein structure. And the cistern breaks up into cargo-laden Golgi vesicles, and then these Golgi vesicles send out for exocytosis their content to the outside of the cell, because we are sending out the material. The process of protein processing and secretion. So post-translational modification is when protein is assembled on the endoplasmic reticulum surface, threads itself through a pore in the endoplasmic reticulum membrane into a cistern. And the enzymes on the endoplasmic reticulum modify the protein, remove some amino acid segments, and fold the protein, stabilize with disulfide bridges. And the protein pinches off from the endoplasmic reticulum in transport vesicle coated with a clathrin protein. This clathrin protein is going to yield in the end to a clathrin coated pit, as we will see in a little bit. And the clusters of vesicles fuse and form a new Golgi cistern known as the cis cistern. The new cis cistern migrates through the Golgi to the opposite side, the trans phase. So the trans cistern breaks up into vesicles containing the product, and some vesicles become lysosomes, others become secretory vesicles that fuse with the membrane to release the content. So it is the endoplasmic reticulum that adds the sequence that allows this material to be exocytosed and then endocytosed on the cis side, of the cistern of the Golgi body. Then the cis side of the cistern of the Golgi body will internalize and bring in all the material, and that is where you're going to have your modifications. And these genes, they can be turned on and off, and the cells can turn some genes permanently off, like liver cells turn off hemoglobin cells, and cells can turn genes on only when needed. So the level of the gene expression can vary from day or an hour to an hour. So this can be controlled by the chemical messengers such as hormones. So an example is in response to prolactin, the mammary gland cells turn on the gene for casein protein only when the breast milk is produced. So casein synthesis and secretion. That shows you an example of gene regulation. So the hormone prolactin binds to the receptors on the membrane of the mammary cell. So the receptors trigger the activation of a regulatory protein known as transcription factor in the cytoplasm. The regulatory protein moves into the nucleus and binds to the DNA near the casein gene. The binding enables the RNA polymerase to bind to the gene and transcribe it, producing the mRNA for casein. And casein mRNA moves to the cytoplasm, and the ribosomes on the rough endoplasmic reticulum translate it. And then after the endoplasmic reticulum bounded ribosomes translated, it will be docked into the rough endoplasmic reticulum, where it's going to receive a signal peptide that allows it to bind and dock into the cis side of the Golgi apparatus, and then the Golgi apparatus secretes it to the trans side of the Golgi complex, and then the secretory vesicles release the casein by exocytosis, and it becomes part of the milk. So that shows you here the gene activation process, where you have DNA codes for mRNA transcription. That's the first step, followed by, in the cytoplasm, mRNA codes for the synthesis of the enzyme. Then the second messenger activates a dormant enzyme. The dormant enzyme converts cholesterol into, and we would have, when testosterone is needed, luteinizing hormone stimulates the production of a second messenger, and that second messenger would bind ultimately. That second messenger would bind ultimately to the enzyme that was generated in the cytosol, and together they are going to activate. They are going to activate an enzyme that would convert for us the raw material. The raw material for all the steroid hormones is cholesterol, and converting cholesterol into testosterone. And from testosterone, we would have, it's going to be secreted. That testosterone is secreted from the cell and exerts various anatomical, physiological, and behavioral effects. Okay, so we will talk about DNA replication. We'll talk about DNA replication next time, and the cell cycle. So this is where we're going to deal with DNA dependent DNA polymerases. So we will do this next week. We will stop here, and